Hello again, everybody. This is Mr. Walters, and we are delving into the nervous system. So, of course, the nervous system can be subdivided into the central nervous system and the peripheral nervous system. The central nervous system, again, is the brain and the spinal cord. The peripheral nervous system, you do need to add this to your notes. It also includes these structures which are coming up here at some point fairly soon. We've got the cranial nerves and the 31 pairs of spinal nerves as the structural component of the peripheral nervous system. Of course, peripheral nervous system also includes the senses, which we've talked about. So go back in your notes from the last lecture and add in the 31 pairs of spinal nerves as part of the structural component of the peripheral nervous system. And there's 12 pairs of cranial nerves. So the cranial nerves as well. All right. Anyway, what we have in front of us here is a neuron. So this is the structural and functional component of the nervous system. Looks quite a bit different from other cells that we've looked at. Cheek cells, for example. Of course, cheek cells have a different role in the human body, so the function is going to be different. Muscle cells have a different role in the human body, and we know that the function is going to be related to the structure, so different functions mean different structures. One thing that's interesting, if you go in here to the nucleus and you analyze the genes present here in the nucleus for this nerve cell, they are identical to the genes found in cheek cells or muscle cells or kidney cells or gastric cells. Those genes are identical. What's going on here is that as development proceeds, a certain set of genes will be expressed through protein synthesis, transcription, and translation. And with those expressed genes, we're going to get a neuron. With a different set of expressed genes, we will get a kidney cell, or a muscle cell, or a gastric cell. So just kind of interesting. Anyway, just checking this out here. We've got the nucleus, so there's the control center. Don't worry about taking notes just yet. Just listen as we go through this. I'll tell you when we get to a different slide what to write down. Notice, so we have the nucleus, the control center. These are dendrites. These extensions here are dendrites. Again, just listen here, just listen. So these dendrites are extensions, and what's going to happen, these dendrites are going to transmit nerve impulses towards this large cell body here. Notice, information's coming in, action potentials are coming in, action potentials are coming in, action potentials are coming in. Here we have the cell body, again the nucleus. We have various cytoplasmic structures, many of which you're familiar with. These are highly metabolic, lots of ATPs required, so these have a large number of mitochondria and many of the organelles of which we've already studied. Notice surrounding this neuron, again, this is the head honcho, this is the big cheese of the nervous system. But this king, so to speak, has plenty of helper molecules. These helper molecules, not molecules, helper cells. These helper cells are neuroglial cells. So here's the nuclei of a neuroglial cell. We'll talk about those in a moment, but for right now, realize they are helper cells for this larger neuron here. If we move down, we have an axon. So axons are going to send out nerve impulses away here from the nerve cell body. So there's the cell body. Axons carry nerve impulses away. So there's a good look at that cell. that neuron, and then notice the helper cells, which are the neuroglial cells. There's several different types of neuroglial cells, which we will talk about here in a moment. Well, just getting to general organization again, general organization. We've alluded to this, but here's a nice diagram which illustrates the general organization. We have the central nervous system here. Now this is important to get, so I do want you, if you have to, pause the video and first of all, actually I take that back, just listen first of all. I'm going to go through it, then pause it, then copy the information here. Notice we have the central nervous system, CNS, and the peripheral nervous system here, the PNS. Notice we have this is a sensory neuron here. So we're receiving information via these sensory receptors here. Now this could be a touch receptor. It could be deep pressure, light pressure. It could be heat or cold. This could be what am I trying to say? This could be a retinal cell. So it could be rods or cones receiving light impulses. Notice these dendrites are gonna carry the nerve impulse information down this axon, and this is the peripheral region here. So what we're doing is we're moving down the pathway of the spinal nerves. So remember, the peripheral nervous system, you've got 31 pairs of spinal nerves, you have the cranial nerves, and you have associated sense structures as well. So notice we're moving down this sensory pathway, and again, sensory pathways are carrying information from the senses to the central nervous system. There is another name for a sensory neuron which I want you to note. So sensory neurons are afferent neurons. Again, just listen now. And of course you can pause the video at any time, but it's more important, what's important I feel, is that you listen first, then pause, then you can go back and take notes. Remember, notes are really important. You're gonna have to recopy those notes. This has suddenly become a very important way of assessment. They have the notes and you recopy the notes. I was moving towards the idea where you were on your own for recopying notes. The fourth quarter was meant to be no more credit for recopying notes. Do it if you feel like you need to. If you don't feel like you need to, don't, because you're getting old enough where you can make those decisions. But people, times have changed. Times have changed. So this is now becoming an important part of the assessment. Handwritten, recopied notes. I need to know that you're listening to these, you're watching these, and then you're writing the notes and recopying it. Be ethical here. Use this time productively and do this. Do this. Be ethical, meaning actually do it. So important. You out there? You listening? Bueller? So we're moving down the pathway here, and there's the cell body of a different nerve cell. We're going to go to the CNS, central nervous system here. So we're going to go to how about the spinal cord here? We're going to send a nerve impulse across from one nerve cell here to the nerve cell of another. These would be dendrites. Dendrites receive, axons send axons away. So these are dendrites. We get the nerve impulse, then we're traveling down this axon, and here we go. So notice now we have an interneuron. So this is an interneuron. Most of the neurons within the spinal cord and the brain are interneurons. So they're receivers and senders. So they're both receivers and they're quarterbacks. So they're receiving impulse, then they're throwing their impulse to another nerve cell. So here are interneurons. And dee 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 dee, that's the sound of a nerve impulse. Dee 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 dee, all the way to here. Now there's a synaptic cleft there. We will be discussing in detail what chemically is going on at these synapses. We've already talked about synapses at the neuromuscular junction. What about these synapses between neurons? This is vital, essential in neurophysiology, that you understand what goes on at these synaptic cleft with neurotransmitters being released and then having an effect on other nerve cells. Just essential. It's essential for how you think. It's essential for how you move. It's essential in certain, when there's certain abnormalities of brain function. It could be to inadequate supply of these neurotransmitters. So far, the only neurotransmitter you're familiar with is really important. It's acetylcholine, and that's the neurotransmitter at all neuromuscular junctions. But now, when we go into the central nervous system, when we're in the central nervous system, there's a wide variety of neurotransmitters, and these neurotransmitters have varying effects on your brain function and your physiology in general. Anyway, so there is another synaptic cleft, and neurotransmitter is going to be released. So again, we're in the central nervous system here. Now the central nervous system is going to enact a response. We're going to send a nerve impulse out, away from the central nervous system, moving in this direction. Now, of course, we're in the peripheral nervous system as that nerve impulse is being sent. This is a motor neuron. A motor neuron, also called efferent. Be familiar with efferent as a motor neuron and afferent as a sensory neuron. We are moving again in this direction away from the CNS, and the action potentials, like a wave on the ocean, that action potential is having its effect down this axon here. And here we go. We are going to move to an effector organ. The effector organ could be a muscle. It could be your bicep, tricep, gastrocnemius muscle, or it could be a gland. It could be the adrenal gland. And this nerve impulse that stimulates the adrenal gland would result in the adrenal gland releasing adrenaline. Can you tell me the release of adrenaline, would that be a sympathetic response or would that be a parasympathetic response? You have to go back and think sympathetic versus parasympathetic. These are two divisions of the ANS, the autonomic nervous system. Which one do you think? Which one is it? Leah, shout it out. You got to be a little louder. I'm all the way at Marquette. Okay, go out to the, go outside. That might help. Go ahead, Leah. You can do this. Yes? Wait a minute. Wait a minute. I think I heard that. You said that that was the sympathetic nervous system. Good job. Sympathetic nervous system. Easy to remember the effects of the sympathetic if you relate the idea of sympathetic with fight or flight. Parasympathetic is rest and digest. Adrenaline is going to be a fight or flight response. So now it would be a good time to copy these notes. Again, pause at your will. No big deal here. We've got the central nervous system, just taking a look here, reviewing the anatomy. These pictures, the information sticks a little bit better in your head here. So notice we have the brain, we have the spinal cord, so that's the central nervous system. We have the peripheral nervous system, which includes these cranial nerves, which run out of the base of the brain It includes 31 pairs of spinal nerves. These spinal nerves are reaching out with their associated axons. Key word there, axons. And they're reaching out, eventually ending where the sense organs are. So eventually, they're going to be receiving or sending nerve impulses to and from the periphery, which could be the skin, could be the eyes, could be the ears. We've talked taste. Any of these senses. Of course, I should say this, if it is the somatic region of the autonomic nervous system, then we're going to skeletal muscles. Well, there are three general types of neurons. Three general types of neurons. One type of neuron are multipolar neurons. Multipolar. Notice these guys have lots of dendrites. These cells have one axon here, so they have an axon here. Notice the direction of the impulse is always going to be down the axons. Most of the cells, and by the way, this says dendrites up here if you can see it. If you can't see it, these are all dendrites. And again, the dendrites are sending, the nerve impulses are moving in this direction towards the cell body here. Most of the neurons of the brain and the spinal cord are these multipolar neurons, and I want you to know that. So examples of multipolar neurons can be found throughout the central nervous system. Let's look here. This is a bipolar neuron. So bipolar. So notice two poles, so to speak, here and here. Here's the dendrite, so we're going D, 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 D. The action potential is moving in this direction. We're going to process this action potential and then send off a response down the axon here. These are bipolar neurons, and the really good example of bipolar neurons are the rods and cones of the retinal cells. Notice here, we've got dendrite. We're sending impulse in this direction. Dendrites are towards the cell body. We'll process that information, then send a nerve impulse down the axon. Notice this is a unipolar neuron, so that's unipolar. These unipolar neurons are found in collections just outside the spinal cord called ganglion, and I want you to get that. So these are collections of unipolar neurons can be found just outside the spinal cord, and what you would have would be a large number of these unipolar neurons, and in these ganglion you'd find a lot of these unipolar cell bodies. All right. I mentioned helper cells. Helper cells of the central nervous system. Just look and listen here. That's what's important right now, just looking and listening. Notice we've got the neuron, and this neuron is important. This is, again, the king right here. This is the king of the cells here. All these other cells are going to be supporting the king. Notice these helper cells, astrocyte, microglial cells, oligodendrocytes. Again, the roles of these are going to be as support cells in various ways, shapes, and forms for this particular neuron. Well, again, you're not responsible for the specific functions of each of these, and I take that back with the exception of these oligodendrocytes. So make a note that the oligodendrocytes are important in producing what's called the myelin sheath. the myelin sheath is essential for many roles in a neuron, of which we're going to talk about in a little bit. So these helper cells, the glial cells, just make a note, they're important in support of the neurons, but make a special note that the oligodendrocytes are important in producing this myelin sheath, which we're going to be talking about. All right, this is a motor neuron. It can be found in your book. One of your assignments is going to be on a nice clean sheet of paper. You are going to draw this motor neuron, and you're going to state a function for each of those structures. Okay? So that will be assigned. I'll put that out on Google. I'll put it on Ren Web. So this would be one of those classic, if you have copy paper, use it. And you'll entitle this the motor neuron. Let's take a look at some of the different structures. I want you to use this lecture, but I am purposely leaving out some information for you to look up and to write in your on your picture. So here we are. We've already talked about dendrites, and dendrites are going to receive nerve impulses and send nerve impulses to the cell body. So that's the function of the dendrites. Notice here's the cell body, which contains the nucleus. Notice we have neurofibrils, we have the axon hillock, we've got the nerve impulse, which is traveling down this axon. If you notice, look at the wrapping. These are those specialized helper cells, the oligodendrocytes, that wrap around the axon and have various roles in terms of importance for this particular neuron. Again, this is a motor neuron. Well, notice the gaps here between these myelinated axons. Again, this is the myelin, and there are the individual cells. Now, along with those oligodendrocytes, we do have Schwann cells, and Schwann cells are also important in the myelination of these axons here for the motor neuron. Notice here we have the synaptic knob, that's the terminal end, and notice the branching that's going on. This one neuron could branch and be communicating with quite a few different structures. They could be communicating with a gland or a muscle, for example. So, for example, each of these terminal ends here could synapse with a region of skeletal muscle, for example. Now, what's the role of the myelination? Notice this particular Schwann cell, it's going to wrap around the axon like a Band-Aid wraps around a finger. That myelin is going to support, so you need to get that function. It's going to insulate, so support and insulate that axon. It's also important in speeding up the conduction of nerve impulses. So nerve impulses are going to be traveling down the axon, and myelinated nerve axons will have a faster transmission of the nerve impulse. If this myelin begins to degenerate from environmental or genetic factors, you could end up with a disease. Multiple sclerosis is a multiple sclerosis is a disease which progressively degenerates the myelination of these nerve fibers. Depending on the severity of the disease, you could end up in a wheelchair or just be a lot slower and have more difficult in using your muscular system compared to before the onset of the disease. Here's another look at the myelin and the relationship between the myelin and the axon itself. So you're not responsible for this diagram, but look at the anatomy. And again, look at the nodes of Ranvier, dendrites, axons, good review there. Of course, nerves need to communicate with other nerves. Just like a nerve communicating with a muscle, when nerves communicate with other nerves, we have these synapses, and the synapse has this gap or this synaptic cleft. So we're going to send a nerve impulse down this particular axon. Notice the Schwann cells, important for myelination. And then we're going to reach this, this terminal region here. I call it the synapse. So you have a region here which is presynaptic, and you have postsynaptic region. So here's presynaptic, here's postsynaptic, and then of course there's the synaptic cleft. Notice we have more synapses here, and this nerve impulse is going to travel here. It's going to have an effect on this region of the associated neuron. And then that information will be processed, and notice we could follow the arrow here, and then we're going to push that impulse down this particular axon. We're going to spend a decent amount of time talking about this region. We could go up here and get a closer look at it. So this region here. So we have the synapse, the synaptic cleft. Notice we have mitochondria throughout. Notice also we have these synaptic vesicles. Synaptic vesicles, if this were a neuromuscular junction, these synaptic vesicles would be filled with acetylcholine, ACH. Again, there is the microscope picture. This is a light microscope picture. And I think right now it's a short break for me, and we're going to get back. The next thing we're going to be talking about is how is it that action potentials, what are action potentials? What are nerve impulses? How are they produced? What are the effects of these action potentials or nerve impulses? We're also going to be talking about the different neurotransmitters besides acetylcholine and what they do. So that's coming up. All right, see you in a bit.